All right, let's talk for a minute about what we're doing in these simulated crosses. What we're going to be doing is a type of linkage analysis. And a couple of notes to begin with for the linkage analyses that we're going to be conducting. In these analyses, in all cases, the wild type is dominant and mutants of various types are recessive. So in these analyses, we're going to be examining what happens to two genes simultaneously to test the hypothesis that the genes are linked. Or actually, we're going to test the statistical hypothesis that the genes are not linked, kind of our null hypothesis, okay? So to start out with, the first cross that we're going to look at is going to be examining a gene for wing length and a gene for body color. So we have two alternatives, and I'm going to use the notation from the simulation. So for wing length, we can have either wild type, which I'm going to symbolize DP plus. Right, so that's the wild type allele, or DP, which is going to be dumpy wings. Right, so that's gene 1 is for wing type. Then the second gene that we're going to look at is for body color. So that is going to be either BL plus, so that's wild type. And the wild type body color is typically a brown color. And then BL, the recessive, is going to be black. Body colors, right? So here's our second trait, body color. And the way that we're going to start this out with our parental generation, we're going to cross a wild type female fly. So wild type female with a dumpy black male, okay? So if we were to write out the genotypes of these individuals, I'm going to write them in two different colors, and I think you'll see why in a minute. If we're writing out these genotypes, this wild type female, she's going to be DP plus, and then she's going to be BL plus, okay? And this dumpy black male, and if we're thinking about chromosomes here, well, we'll get to that in a minute. This dumpy black male, right, is going to be DP, BL. So if we're going to go from this generation to our F1 generation, the next generation, right, first thing we always need to do is think about gametes. And notice, so every gamete for this wild type female needs to contain one allele for wing type and one allele for body color, right? So it doesn't make sense to only have a wing type allele or only a body color allele. They need both traits. So for this wild type female, there's only one possible type of gamete that she can produce when we're just looking at these two genes, right? All of her gametes are going to be DP plus, BL plus, okay? Similarly, for this male, all of the gametes that he can produce are going to be identical, right? So he's only going to produce DP, BL. So let's just think about this from the standpoint of if these are linked, that means that they are physically on the same chromosome, okay? And so now, if these come together, our next F1 generation of flies are going to be DP plus, BL plus, right, from their mom, and then they're going to get DP, BL from their dad, right? So there's one homologous chromosome. Here is the other homologous chromosome. Now, if we want to test the hypothesis that these genes are linked, what we need to do is conduct a test cross by crossing these offspring with a homozygous recessive individual because that's going to give us a very useful predicted ratio of offspring in the F2 generation that's going to let us distinguish between genes being linked or genes not being linked. So we're going to cross this individual with another one who's homozygous recessive, DPBL, DPBL, right? So when conducting this cross, the next thing we need to do is think about the types of gametes that an individual can make. So if you look on page 6-21 up top, we're first going to look at predictions for dependent assortment. So these genes are completely linked. If these genes are completely linked, when this individual goes through meiosis, she's got one chromosome from her mother, one chromosome from her father, so she's only going to be able to make two types of gametes. So I'm going to just note here, if 100% linked, female gametes are going to be only DP plus BL plus and DP BL. And then from the second parent here, right, they can only produce one type of gamete. They can only produce DP BL. So if these are completely linked, I'm only going to wind up getting two possible combinations. So instead of having a Punnett square, I'm just going to have a Punnett rectangle, right? So these offspring are going to wind up having DP plus BL plus, right, from one chromosome from the mother, and then DP BL from the father. And in the second box here, we're going to wind up with DP BL from the mother and DP BL from the father. So that's our genotypes. And what about our phenotypes? So these individuals are heterozygous for both wing type and body color. And so because they're heterozygous, they're going to wind up having the wild type phenotype. And then in the second box, these individuals are going to wind up having the dumpy black phenotype. All right, so that's if those genes are 100% linked. What about if the genes are unlinked? In other words, they're on separate chromosomes. Right? So that's going to look something more like, here's my DP plus, here's my BL plus, and the mother. And again, this is sort of arbitrary that this is mother and that this is father. Right? And then the mother, she's also going to have a DP and a BL. Right? So they're separate chromosomes here. And same thing crossed with a homozygous recessive individual. Those are unlinked. So again, next thing we need to do is think about what types of gametes could be produced. And now if these are unlinked, we've got independent assortment to think about, right? So it could be that these top two alleles, wind up in a gamete together. It could be that this allele, right, those two could wind up together. It could be that this DP plus allele winds up with a BL allele, right? That's equally possible if those chromosomes just independently assort in the opposite direction. And then same thing for this DP allele. The DP allele could wind up with a BL plus allele, or the DP could wind up with the BL allele. So we have 4 types of gametes, right? So let's just write those out. So we're going to have DP plus BL plus, right? Could also have DP plus BL. Could have DP BL plus, and we could also have DP BL, right? All of those are possible combinations if unlinked. Now, what about the second parent? Well, look, DPBL, DPBL, those are the same thing. So this parent still can only make one type of gamete. All that it can make is DPBL, 100% DPBL. So now, if I am going to draw this out as a Punnett square, it's actually more like a Punnett rectangle again. If these genes are unlinked, now there are 4 different possible combinations I could wind up with, right? So I could wind up DP plus, BL plus, DP, BL, and actually across all of these, right, there's always going to be a DP, BL from the father. Right, and so then the pattern here, so this is going to be DP plus BL. Here we've got DP, BL plus, and last but not least, DP, BL. So that's our genotypes, but what about our phenotypes in this case? Well, this upper one is going to be wild type. How about here? That's going to be wild type for wing type, but then it's going to have black body color. So I'm just going to say wild wings, black body. Here we've got dumpy wings, wild type body. So dumpy wings, wild body. And then down here we're going to have dumpy wings and a black body, okay? So those are our 2 contrasting scenarios, right? So if these genes are 100% linked, we only expect to see 2 phenotypes, wild type or dumpy and black, the parental phenotypes here. If those 2 genes are unlinked, then we wind up seeing 4 different possible phenotypes instead, right? So 2 of those phenotypes match up with a parent up here, but 2 of those phenotypes do not match up with the parents. For our linkage analysis, we're going to be working with virtual flies. So to access the website with virtual flies, you're just going to go to mynewbite.com. So mynewbite.com. When you get here for a login, for your email address, you're going to use studentsc at newbite.com, and the password is studentsc. Once you're here, you're going to click Biology, Drosophila Genetics Lab, and we're just going to use this top one here. This will bring you to a welcome page. Just click to continue. And for this exercise, we're going to use the button that says Calculation of Linkage Distance. We're going to be examining linkage between 2 different pairs of genes. So first the dumpy and black genes, and after that the purple and black genes. So from here you're just going to click on dumpy and black, and then click OK. That's going to bring you to your fly window here. So we're going to pull open the lab book from the left-hand side, and then we're not going to work with a lab book here. We're going to work with the material that's in our lab book, but we are going to use the counters that are in this lab book. So I'm just going to bring up the counters, and then I'm going to move them down to the bottom of the window so I can see what I'm doing. So the first step is just to examine the genotypes of the parent flies. And for working with this, there's a couple of different ways to designate different genotypes. So we're going to change our genotype display in this case to the display that's listed here as VG plus VG, so that notation for our alleles. So then I'm going to come over to the parent vial, and I'm going to examine the female, the mother fly, just by dragging her into the observation window. And what you can see here is that she has the wild type phenotype, so she has long wings, and she has a brown body. But down below we can also see her genotype, so she is DP plus BL plus DP BL for her genotype. So I'm going to put her back in the vial, and let's take a look at our male fly. So a male fly here has a black body, and he has dumpy wings. So notice his wings don't stick out very far past his body. So that's our other phenotype that we're working with. So now that we've taken a look at them, we're going to breed them. So we're just going to click the arrow to breed. And now we've got a whole bunch of fly offspring. So I'm just going to take one of those offspring, bring it up to the observation window. And so in this case, the phenotype of this fly is dumpy and black. So I'm going to come down to the counter and I'm going to click 1 for dumpy and black. And that's going to automatically bring the next fly up into my counting window. So in this case, I've got one that is dumpy but not black bodied. So it just goes in the dumpy offspring. Here's one that's a wild type. Here's one that's dumpy, wild type. Here's dumpy and black. And here's one that's black but has long wings. Okay, so you get the idea here. So we could sit here counting up flies. Here's black, wild type, dumpy and black, etc. So we could spend a bunch of time doing that, but we've got other things to do with our day. So what we're going to do now is go up to the Options tab and go down to the bottom here where it says Auto Count 10,000 Flies. If you press that, that's going to open up a window. Note here it says flies left in the offspring jar are also counted and removed. So here is our auto count of additional flies. If I press OK, that's going to be added onto my counters down at the bottom of the screen here. So this is the information that you're going to need to fill out the table that's on page 6-23 of your lab manual. Alright, so let's say you've gone through, you've run your simulations, you've counted your flies. Couple of things to notice, right? So you're going to use that auto count 10,000 flies. Your numbers are probably going to be different from my numbers, okay? So just check that. The other thing I want you to notice is that the phenotype order here is different from the order in this simulation, right? So this is organized by wild type, black body, dumpy wings, wild type body, dumpy wings, black body. So just make sure you make note of that. So you're going to need to count up the total number of flies of each type, get a total, look at percentages, then answer these questions about these predictions based on whether the genes are completely linked or unlinked. Okay? Once you've got all that, then you're going to be set up for some of the chi-square analyses we're going to do for this part of the lab. All right, so here's the spreadsheet that we're going to use for our analyses for this lab. Note that in the first two tabs, serrate and glass, we didn't tabulate everything into the spreadsheet, so you're just going to have totals here. So take the totals from your lab notes, copy them in. Note what percent are the mutant phenotypes, and make a note about whether serrate seems to be a dominant or recessive trait. From there, the next thing you're going to do is conduct the chi-square analysis to determine whether the observed numbers of flies that were either wild type or serrate match up with what you expect based on the serrate allele being either the dominant or the recessive allele. So you're going to fill out this table down here, first with your observed number of flies. Use that to calculate your total number. Use that to calculate how many flies you'd expect of each type. And remember, this should be calculated as a number. Work through these calculations and you'll come up with a total chi-square value. Here we have two categories, so we're going to be working with 1 degree of freedom, so you're just going to look across the top of this table to figure out what your p-value is, and that's going to tell you the probability that what you observed actually matches with what you expected. Okay, so if you have a high probability, there's a good chance that what you observed matches with the pattern you expected. If you have a really small p-value, a low probability, that means that there's some kind of difference between what you observed and what you might have expected based on that pattern of inheritance. So there's another tab here for the glass-eyed flies, so same process for those. The last one to look at is this tab for the linkage analysis. Let me make this a little bigger. So again, a very similar process here. Some of the titles got cut off in the rows, so I just reached up here between columns B and C and just dragged across so we can see that better. But we're going to go through the same kind of process. So for example, for my wing and body color analysis, I had 3,235 that were wild type, 1775 wild type black, 1809 that were dumpy wild type, and then 3280 that were dumpy and black. I'm going to use a function equals sum to add all of those up, and then we're going to do this based on the expectation that these genes are unlinked, right? So coming back over here, that's the expectation that we're going to get equal proportions of these four phenotypes. So I'm going to use that to calculate my expected fly numbers and go through and complete the chi-square analysis. So the next thing we're going to do is work through how to construct a linkage map. So this first involves obviously conducting a ton of crosses to look at the percent of recombinants between multiple different gene combinations. So for the purpose of this example, I'm going to show you a collection of crosses, between a set of different genes here. So it's different from the genes that you're going to analyze, but will help illustrate the major point. So for constructing a linkage map, what we want to do is we want to start out by looking at the genes that are the furthest apart from each other. So in this case, those genes have a recombination rate of 50%. So we're going to start with sepia and mahogany. So I'm just going to draw a line for my map. And I'm going to put sepia over here and mahogany over here. Okay, so I've got those in position. And the next thing I'm going to do is I'm going to start looking at other genes in between sepia and mahogany. So let's take a look at the next furthest apart genes, which are the genes dikete and mahogany that are 47% or 47 linkage units apart from each other. So the thing about this is, right, dikete could be on either side of mahogany. It could be here between sepia and mahogany, or it could be out here, off the chart here somewhere. How are we going to be able to tell? The way we're going to be able to tell is if we also look at the relationship between dikete and sepia at the same time. So notice that dikete and sepia are closer together than dikete and mahogany. So that tells me that dikete has to be in between sepia and mahogany. It can't be out here, because if this number were also 50%, then I'd know dikete was somewhere out here. We don't really know where it is. But in this case, between those two pieces of information, we can figure out that dikete is located in between sepia and mahogany. Okay, and now more specifically here, we have a recombination rate for dikete and mahogany of 47%, so that's going to translate to 47 map units. And for dikete and sepia, that recombination rate is 15%, or 15 map units. And now notice something. 47 and 15 add up to a number that is greater than 50%. So as it turns out, sepia and mahogany, we can now add these up, right? So that's going to give us 62 map units. Sepia and mahogany are 62 map units apart. So you might wonder, why do we get a recombination rate of 50% for sepia and mahogany if in actuality there are 62 map units apart? And that's because 50% is the maximum percentage of recombinants that we could possibly get using linkage analysis. So that's why we have to work with multiple genes to really map out positions. So now I've got dichete and mahogany accounted for, dichete and sepia accounted for, sepia and mahogany accounted for. So we're ready to move on to our next set of genes. So up next we've got this stubble gene. And so what we notice here, so dichete and stubble are 17% apart. So that could mean that stubble could be down here, 17 map units away. Or stubble could be over here, 17 map units away. What do we notice about the next entry, sepia and stubble? Well, sepia and stubble are 32 map units away. If stubble were over here, sepia and stubble would only be 2 map units away, not 32. So that tells us then, okay, stubble has to be on the other side of dichete. It's over here. So we can draw in stubble somewhere like right here. And we know that we've got 17 map units here. And then we can actually check our work. Sepia and stubble are 32% recombination, 32 map units apart. We've got 17 here, 15 here. That adds up to 32. So yes, that corresponds with what we would expect. And then we've got one last check here, which is we also have recombination rates between mahogany and stubble of 30%. So we already said here that so mahogany and dichete are 47 map units apart, right? Okay, so what about mahogany and stubble? Well, 47 minus 17% does give us 30 map units here between mahogany and stubble. And so through this whole process, I've now been able to map out the order of these 4 genes on this chromosome, and including how the relative distances between them based on recombination rates.